Hello everybody, my name is Dr. Vance Trudeau from the University of Ottawa in Canada's capital city. I'm going to talk to you about hormones today, in particular in amphibians. So I just wanted to introduce my lab and then introduce some general concepts for those of you that are new to endocrinology. So let's start with the laboratory. So I'm interested in hormones that control reproduction, sexual development, and metamorphosis. In particular, how the brain and pituitary regulate reproduction. This is called neuroendocrinology, or the neuroendocrine control of reproduction. In particular, in fish, we've used some of these concepts to understand spawning in frogs. And the intersect between these two, of course, comparing fish and frogs, gives us insight into evolution of hormone systems. Understanding sexual development and metamorphosis is really important for captive colonies and captive breeding, because you need adequate sexual development and normal metamorphosis to get healthy individuals for your colony or for your reintroduction. So we've looked at sex hormones, testosterone, estradiol, those kind of things, and the thyroid hormones, T4 and T3, and how they regulate metamorphosis. Now, these processes are very susceptible to upsets through pollution. So we've also looked at endocrine disrupting chemicals, EDCs. And this is very important to understand as well, especially when you're considering a particular site for reintroduction. It might look good. Make sure that it's not a polluted site and you end up disrupting normal development in the introduced tadpoles, for example. So we're open for collaborations, and do not hesitate to email me with questions or what have you. I will be on holiday when this is presented to you, and so please enjoy the workshop, and I hope what I provide you today will get you excited about endocrinology and how we can use it for amphibian breeding. So what is endocrinology? And how can we use it to improve art? Very simply, endocrinology is the study of hormones, and hormones signal and give messages to different parts of the body to control various processes, be it development and metamorphosis or spawning in the reproductive season. So how can we use it to improve amphibian reproductive technologies, and where should we go in the future with our research? So I'll give you a couple of definitions, and then we'll move into some specific examples of hormones and how they have been used in reproduction in amphibians. Okay, so endocrinology is a subdiscipline of physiology, and is concerned with the study of chemical messengers, known as hormones. Now, hormones are a wide variety of molecules, very different categories of hormones. They're secreted traditionally from endocrine tissues. These are the ductless glands, like the pituitary gland or the testes or the ovaries. And the hormones released travel in the blood to eventually alter the activity of another organ. So in the case of the pituitary gland, the hormones act on the testes or ovaries to cause secretion of another hormone, the sex steroids. This must be contrasted to exocrine glands that secrete substances into ducts. So these are the traditional definitions, and these have changed a lot over the last 120 some years, when Starling, in his famous lecture to the Physiological Society in London in 1905, where he introduced the word hormone, derived from a Greek word meaning I excite or arouse to activity. So what is an example of a ductless gland? The pancreas is a combination of ductless glands and exocrine glands. So insulin would be secreted from the endocrine part of the pancreas, and the digestive enzymes would be secreted into ducts to help us with digestion. So these are two types of secretion. One is endocrine and one is exocrine. We're concerned with endocrine tissues when we're talking about induced spawning and captive breeding. Endocrinology is the study of chemical communication then between cells. This can be in normal situations or in pathological situations. For example, there's endocrine tumors and hormone-dependent cancers, breast cancer, prostate cancer, for example. These are situations where the chemical messengers, the hormones, are upset. So hormones can change development or a physiological response. So in the case of sex hormones, you can change the sex of an animal, the sex of a frog, the tadpole, or you can do a physiological regulation. Okay, so hormones can be homeostatic in nature, so controlling balances in the body, or control growth and differentiation, for example, during metamorphosis or development of muscle or what have you. So endocrinology then is the study of integrated physiological responses. The nervous system, the brain actually controls spawning. Immune cells release factors that are considered hormones. The cytokines and the interleukins that you may know from cancer therapies are actually important hormones as well. And of course, the typical endocrine systems, the pituitary gland, the thyroid gland, the pancreas, the testes, and the ovaries. These are all considered endocrine system. And they're not separate. They can interact and communicate with each other. And this is a very important concept in integrated or integrative physiology, if you will. So here's just a simple diagram to show the traditional definition of endocrine secretion. So here's cells in an organ. These little red dots are secretory vesicles. So these would be a peptide or a protein released from the cell and goes into the blood vessels and be carried somewhere else. So this could be the pancreas releasing insulin, and insulin goes into the blood and acts on muscles to allow glucose to be taken up and regulate your glucose levels. So this is a very simplified version of endocrinology, but there's other types of communication that fall under the term endocrinology. For example, paracrine, para meaning beside. So one hormone from one cell can act on an adjacent cell. And this is really important during development and especially during metamorphosis in frogs. One cell in the developing legs can influence another cell through growth factors. Now, my specialty is neuroendocrinology. Neuro, so you need brains and neurons, and you need hormones. So there are hormones in the brain, especially in what's called the hypothalamus, that will produce peptide hormones, and they are released into the blood vessels of the pituitary gland and affect pituitary gland function. So a neurohormone can be secreted into the blood as well. So you may know the neuropeptide called gonadotropin releasing hormone, GnRH. It's produced in neurons, so in the cell body, transported along the axon, packaged into secretory vesicles, and upon stimulation would be released into the pituitary blood system and then delivered to the cells that control spawning. And we're going to go into that in much more detail. There are many types of hormones. You may be familiar with the steroids. The first one discovered was ecdysterone, which causes molting in insects. But you're probably familiar with testosterone, dihydrotestosterone, estradiol, progesterone as the sex steroids. The stress hormone cortisol is a steroid. And vitamin D is actually an important hormone for controlling calcium levels and bone health. So the sex steroids are derived from cholesterol and converted to all these various forms. Thyroid hormones from the thyroid gland, they contain iodine, so there's thyroxine with 4 iodines, T4, and triiodothyronine, T3 with 3, and this is the biologically active one, controlling metamorphosis, for example. There are many protein hormones, insulin, growth factors, and the pituitary hormones, and we're going to go into the details of some of the pituitary hormones, which are really relevant for today's discussion. Peptides are also very important. They're really short. For example, gonadotropin releasing hormone is only 10 amino acids long, but it's necessary for reproduction. Oxytocin causes milk release or important in birth in mammals. It's only 9 amino acids long, and there's many, many others, and we won't cover them today. I already mentioned the cytokines and the interleukins. These are from immune cells, but they are chemical communicators and hormones as well. There's so-called non-classical hormones, for example, neurotransmitters. We will talk about one of these today called dopamine. So let's start with the hormones of the anterior pituitary. The first family, important for today's presentation, is the growth hormone and prolactin family, GH and PRL. So growth hormone, as the name implies, stimulates growth. You can do this both directly and indirectly. So as when you're a child, you grow from the tips of the long bones, called the epiphysis, and a substance called chondroitin sulfate is added for growth. And growth hormone stimulates these cells, the chondrocytes, to produce CS, and that helps you with bone growth. So growth hormone acts directly on these cells. Growth hormone is anabolic, so it can stimulate amino acid uptake and incorporation into muscle protein, and it helps with collagen deposit. So growth hormone, or things that can increase growth hormone levels, are used illegally by athletes to increase their muscle protein and strength and what have you. Actually, many of the growth-promoting effects and other effects of growth hormone are indirect through the liver. They act to stimulate another hormone family called insulin-like growth factors. So they act like insulin, but they don't look like insulin, and they're super important for growth. So IGFs are produced by the liver upon stimulation by growth hormone that comes from the pituitary. The IGFs are then released into the blood to affect tissue growth. So here it is kind of illustrated here. Growth hormone from the pituitary can be released into the blood and act on bone growth. So it's really important during annual development to get a good calcium incorporation into bone and make sure that your metamorphs are happy and healthy. Growth hormone can act on the liver. Then the liver produces insulin-like growth factors. So the liver is an endocrine organ, not only a metabolic organ as well. IGFs then act on muscle and other tissues to affect growth and other processes. So growth hormone has many roles, and I'm just going to cover a couple today. They have some role in ionic balance in fish, helping salmon, for example, go from freshwater to saltwater. And growth hormone can act on the testes or the ovaries to help with the production of sex steroids. So growth hormone works along with the gonadotropins to help in particular with estrogen production. Prolactin, you can guess that it's really important in mammals because it's involved in mammary growth and development and the production of milk, called lactogenesis. And it usually works with other hormones to stimulate complex physiological processes. So estrogens and prolactin can encourage mammary growth. So prolactin and insulin can affect metabolism. Glucocorticoids and progesterone and things like this can interact and help with prolactin's action as well. So these are typical mammalian responses. And in terms of reproduction, in particular in rodents, prolactin is known to be luteotrophic. So it acts in concert with the gonadotropic hormones from the pituitary to stimulate progesterone synthesis in the corpus luteum of the ovary. So when a mammal ovulates, releases the egg, the cells left behind change how they produce hormones, and prolactin can help with the production of progesterone, which is needed for development of the uterus of the pregnant female mammal. So this is a mammalian example. It doesn't have much relevance to amphibians, but it's the classic example of how prolactin works. Prolactin may also regulate testicular function in males, helping to induce receptors for the gonadotropins, in particular LH. So why is this important? Because prolactin can assist LH in regulating steroid production in the testes in particular. So the big question, is prolactin found in non-mammals? And absolutely it is. It's in all vertebrate species. Now in birds, prolactin is really important for stimulating maternal behavior and brooding behavior. So it has an important role in reproduction. Prolactin is also a growth hormone in anurine tadpoles. So it acts with growth hormone to regulate growth of the tadpole. Now, what about a reproductive role in amphibians? So the best data comes from a lab in Japan studying the Japanese newt. So prolactin from the pituitary, it's released into the blood but gets into the brain and can stimulate male newt tail vibration behavior, which is important for courtship. The synthesis and release of the female attracting pheromone sodofrin, which is a peptide, is produced from the abdominal glands, released into the water, and the male red-bellied newt uses this to attract the female, and this is under the control of prolactin. So here's a picture. Here's the male. He's pushing water towards the female here, and then he's going to switch around and show his abdominal glands and release the chemical sodofrin, a sex pheromone, attracting the female for breeding. So prolactin is really important for sexual behavior. Now, the glycoprotein hormones are really important for today's discussion. There's three of them: thyrotropin, TSH, follicle-stimulating FSH, and luteinizing hormone, LH. So these are in the same molecular family because they have similar structures, and I'm going to go through that in a second. And it's really important to understand the structures, because then you know why sometimes some preparations of gonadotropins actually don't work, okay, to stimulate breeding. So TSH stimulates the synthesis and release of the thyroid hormones from the thyroid gland. It stimulates T4 production, and then T4 is converted to T3, and T3 then regulates metamorphosis. So TSH, because it stimulates thyroid gland development and thyroid hormone release, is really important for metamorphosis. So your environmental conditions must favor growth in some phases for tadpole health, and another time must favor thyroid gland development and thyroid hormone production for adequate metamorphosis. FSH, as the name implies, is important for follicular growth in female mammals and frogs too. It has a role in spermatogenesis in males. It acts along with LH to stimulate sex steroid production. LH drives the process. FSH can help steroid production. Now, luteinizing hormone, LH, is really the key regulator of spawning. It's the surge of LH that causes ovulation and changes in steroid production that are really important for successful breeding. Okay, so it stimulates sex steroid production in the ovary and testes, and it stimulates ovulation or egg release from the ovary in females. So with induced spawning, we hope to induce an LH surge, which is a rapid, huge increase in LH that actually causes the final oocyte maturation and then the release of the egg or eggs. So these two hormones, FSH and LH, are collectively known as the gonadotropins, gonadotropins, or gonadotrophic hormones, depending on which country and which spelling convention you use. But they're basically the hormones from the pituitary gland that act on the gonads to stimulate their development and eventually to stimulate steroid production or the release of the ovary or the sperms. So each of these hormones is composed of two chains linked by hydrogen bonds. There's an alpha chain and a beta chain, and they're 90-something to 100 and something amino acids long. These are different proteins from different genes that are made in the cells of the pituitary gland and then assembled to make active hormone. So once they're assembled, you have to add carbohydrate, and the carbohydrates increase solubility for secretion and increase the half-life in the blood. So this makes it biologically active. So this is really important, and one of the limitations with using recombinant protein technology to try to develop amphibian-specific gonadotropins. They don't exist until now, because this step is really hard to recapitulate in vitro, and we don't have any synthetic gonadotropins for amphibians yet. Be a great PhD project. There is a common alpha subunit that would combine with one of the beta subunits. So there's 3 different beta subunits. There's the TSH beta that would combine with alpha to give you mature TSH. The FSH beta with alpha gives you mature FSH. LH beta with alpha gives you mature LH hormone. So in the jargon of an endocrinologist, we say that the beta subunit confers hormone specificity. That is to say, the specific beta subunit gives us the specific effect of TSH that is very different from LH or FSH, for example. So here's an alpha and a beta. They join up, and I said this already, but you need an alpha with a beta to get the mature hormone, and then add on the carbohydrates in a specific way to make it biologically active. So when you're stimulating the pituitary, you have to give it time to be able to do this. So in your protocols for induced spawning, you have to consider the synthesis of the gonadotropins. Sometimes you have to encourage more synthesis before you try induction, and I'm going to show you an example of that later. So here's a figure from our paper in 2016, which shows some aspects of the control of LH and FSH release. And I'm going to go into this in much more detail, but what I wanted to illustrate here is that the particular part of the brain called the hypothalamus releases factors that we'll talk about later that controls LH and FSH synthesis, assembly, carbohydrate addition, packaging, and eventually release into the blood that will then control steroid production, progesterone, testosterone, E2, for example, and the testes or the ovaries. That's really important for courtship behaviors and liver function and what have you. So one very old and in some species successful method is the use of human or horse gonadotropin preparations. So you simulate this release by injecting something called human chorionic gonadotropin. So this one is produced by the chorion of a pregnant woman. It's secreted into the urine, can be collected, and can be used for induction of spawning in xenopus, some bufonids, and the clulos have shown that it can work in an Australian myobatrachid. Rarely works in ranids, even at extremely high doses. Some people have used pregnant mare's serum gonadotropin, so in equids as well, there's a gonadotropin produced that can be harvested in the urine and used for treatments. But it's not widely used. These are not very stable once diluted, hard to keep, you need freezers and all that kind of thing. So for some species it works. It's the traditional way to stimulate breeding in xenopus, for example. So where I'm going to go in a moment is to tell you how to trick the animal into releasing its own gonadotropins, which will then induce spawning. Okay, I want to get into a little bit more of the gonadotropins in the next 2 slides. Okay, so we did a phylogenetic analysis of the LH beta subunit, the FSH beta subunit, and the chorionic gonadotropin, so the HCG or PMSG. So here is the result of the available data at the time, a couple of years ago, and you can see very clearly that all of the different FSHs from all of the different vertebrates grouped together, and all the LHs grouped together. So from an ancient genome duplication, these two different molecules evolved, and very recently there's an LH-like subunit that was formed during evolutionary time over here, which is the chorionic gonadotropin. Okay, so this is only distantly related to LH beta. This is one of the reasons why it doesn't work very well in a lot of frogs. It's not a perfect mimic. It's a very useful tool, but there's other ways to go about it. And Paul Licht, very famous comparative endocrinologist working in amphibians and reptiles, showed already in the 70s that HCG does not bind to RANID LH receptors. He used a couple of different RANID species to show that it actually doesn't bind very well to the receptor. So it should not work very well. That's what I'm trying to say. Now, the crystal structure of the human gonadotropins has been produced. And this is very interesting for those that like biochemistry, but I just want to re-emphasize how different the LH is from the chorionic gonadotropin and from LH. So in the blue here, we start in the middle with LH. It's the beta chain is here. It's folded in a complex way, and the alpha chain in the green folds onto itself, and they are anti-parallel, and they align, and then they're going to interact and join and form the complete biologically active hormone when the carbohydrates are added. So this is really important to maintain this structure. So any kind of storage of these types of hormones really needs -80 freezers and what have you. Here's FSH, slightly different, well, quite a different structure. Again, you have your beta chain and your alpha chain. Chorionic gonadotropin kind of looks like both of them, where you have a very different structure. So those of you that like this kind of information, you can look at this paper from 2017. There's many other papers about structures of the gonadotropins. So one area of investigation would be making recombinant amphibian gonadotropins, making sure they fold well, and using them for spawning induction. We've tried it, but we did not get a biologically active form of LH, unfortunately. So the last part of the talk is really directed at another type of way you can do spawning in amphibians. There's a couple of nice little pictures here, and really about, I don't know, 15 years ago, we were doing work on ecotoxicology and needed leopard frog eggs at a specific time in development. So rather than collecting the wild ones, which is problematic, you don't know their history or what have you, I wanted to induce spawning in the leopard frog. But I didn't want to spend 15 or 20 years doing the basic research. I wanted to do some educated guessing, looking at other species, fish in particular, trying to develop a new method for spawning induction. So we started with the northern leopard frog, Lithobates pipiens, the problem species. It does not breed in captivity until we developed our methods. So why did I start with this? Well, we were working on them. It's widely used in research, especially for before Xenopus really took over. For example, John Gurdon, first clonings was done with leopard frog tadpoles. Professor Nace of the University of Michigan in the 60s tried to set up a colony. So she was really good at growing tadpoles, and a lot of the protocols are still in use. But in the original paper, they could breed 1 or 2 animals, and they didn't say anything about it. And the colony failed because leopard frogs don't breed in captivity. So the so-called iconic frog that was in high school dissection labs, university dissection labs, used for development, all have been declining, and NACE already recognized this in the eastern U.S. in the 60s. So how do we get them to breed? So I looked at my work and other people's work in teleos reproduction and found some old papers on Rana temporaria and Rana pipiens from a group in Poland and working with Professor Licht. So here's the situation in the goldfish. This is a pituitary gland. These are very small forceps. You can see it's very small, but it has all of the hormones you need. So this represents the pituitary gland here, and as I told you, the gonadotropins are released, acts on the gonads to release sex steroids. So how can we do this? So the principal driver of reproduction is considered to be the neuropeptide GnRH, gonadotropin releasing hormone. So that will stimulate LH and FSH. Now in fish like goldfish and carp, this does not work very well because there is a strong inhibitory system based on the neurotransmitter dopamine. I'll abbreviate it DA. So dopamine is the brakes, GnRH is the gas. Green is go, red is stop. So we have to manipulate dopamine, and in fact, in goldfish and carp, you have to block dopamine before GnRH will stimulate spawning. Now, early reports in Rana temporaria, doing brain lesioning, injecting various dopaminergic compounds, were very, very suggestive of inhibition of dopamine. In fact, in one paper, they block dopamine and can get ovulation in a hibernating Rana temporaria. Very strong evidence that dopamine is inhibitory. What about the GnRHs? So it was found that GnRH is now 50 years old. Its discovery in mammals produced mammalian GnRH. Okay, 10 amino acids long. And now we know that there's many different forms. There's 2 forms in frogs. They're categorized into different types of molecules. So GnRH1 is what everybody has been studying. GnRH2 is the most widely distributed. So frogs have GnRH1 and GnRH2. Salmon and other teleosts have GnRH2 and GnRH3. And lampreys have all kinds of very weird-looking ones that's categorized in the GnRH4 family. So it's really important to understand the biochemistry of GnRH and which one you're injecting. Now, a lot of you will just buy LHRH off the Sigma shelf, and what you are buying is a GnRH artificial form, a synthetic form. LHRH was the original term, and it is 50 years old. It's only 40 years out of date. So please call it what it is, which is gonadotropin releasing hormone, and it's really important to understand which one you're handling. Only one type has ever been tested to any extent, and that's the synthetic version of mammalian GnRH called LHRHA or GnRHA. So please be careful with which one you're testing. I would like to talk about another example, the opposite of the leopard frog. This animal, the wood frog, breeds very easily. So we can use it as a model to show a little bit more about dopaminergic inhibition. So wood frogs in the breeding season, they're explosive breeders. In the lab, if you put male and female together, in a few minutes or a few seconds even, they go into amplexus. So how can we inhibit that to understand what controls spawning? So here are different treatments and different parameters we measured with in the wood frog. Control, a dopamine drug called quinperol injected into females only. So the females would have the dopamine drug and the males would have control injection. then males injected, females not, and then both injected with the chemical. This is the number of pairs in amplexus, and you can see that most of them go into amplexus. The median time in minutes to amplexus is quite variable. Mostly these are very delayed. And what's really important is the median time to ovulation. So we can delay ovulation with dopamine. So maybe if you take away dopamine, you can advance or at least control ovulation. That's the idea. So here's the data. When you put them together, they will ovulate and oviposit, in fact. So this is oviposition time. And you can see here it takes between 160 and 960 minutes with a median time of 690 minutes. So 10, 11 hours kind of thing. And in controls, 8 out of 8 will lay eggs with no treatment whatsoever. Inject females, only 2 out of 8 will lay eggs, and they lay them very, very late. If you inject the male, there's not much effect here. 8 out of 9 will lay eggs, and it's not delayed. When you put the two together, it's again 2 out of 8 will lay eggs, and when they do, it's extremely delayed. So you can, you know that dopamine then controls time of spawning and actually egg release. Now let's go into a little bit of the GnRH biochemistry, and like I said before, this is really important to understand which GnRH you're using and is it the best one. This is a wide open area of investigation in amphibian reproductive biology. There has essentially been many papers using GnRH, agonist, and it's always the same one. Lots of reasons to think that there's other ones that might be better. So here's a simple illustration of the structure. The N and the C terminal with 10 different amino acids. Now the first one to ever be changed and make a synthetic version is the D-ALA6 change at position 6. So when you buy LHRHA from Sigma, you're buying the D-ALA6 GnRH agonist. Now this is a good one. It's gone generic. It's relatively inexpensive compared to some of the other GnRHs. But my question is it the best one? And I don't think it is, because it doesn't always work. So we can think of different projects to start testing different GnRH forms that might be better in amphibians. So keep that in mind. If somebody wants to get into this, I'd be happy to discuss projects with you. Now, dopamine is a catecholamine neurotransmitter. Here's its structure. And to stop dopaminergic inhibition of spawning, then you have to give something called a dopamine antagonist. This will block the effects of dopamine. Now, there's a lot of pharmacology known about dopamine because it's important in all kinds of clinical problems. There's many functions in the human brain, frog brain and fish brain, and it's very important for the control of reproduction. It inhibits LH release in teleos, frogs, rabbits, sheep, and human. So the idea then, if it does that, then maybe it is a likely inhibitor of spawning in your particular frog. So we can use dopamine antagonists to block dopamine. Now the one I chose is called metoclopramide. It is generic, very inexpensive, and it's water soluble, so it's easy to handle. A lot of dopamine antagonists are not water soluble, so you have to give nasty solvents to dissolve it. So it's not the easiest way to go. So I made some educated guess based on this conserved mechanism through GnRH and through dopaminergic inhibition to develop a spawning method for leopard frogs that don't spawn in captivity. Okay, so basically you take the agonist and you're going to inject it, but you have to get rid of the dopaminergic signal. So you have to give an antagonist at the same time. So if you think of this, GnRH will give you a little bit of LH release, but not an LH surge. But if you put an antagonist, you get more LH release, you cause the LH surge, and you get sperm and egg release. And this is exactly what was done in fish. The choice of your GnRH and your antagonist is what differs from the fish method. And it works really well in Lithobates pipiens. Our very first study, we got 100% spawning with high fertilization rate. It was a very dry year, so the next year was a bit problematic. Animals weren't in very good condition. We collect them in the wild and try to breed them in the captivity, but we still got 60%, and at the time, this was the best result in the published literature. We also did artificial hibernation in the lab out of season, getting 2 out of 8 pairs to spawn. So this has been vastly improved, as I will show you. And it works in many other species. So the original paper, you can consult. It's open access online. That paper and the second paper is where everything is described. So we came up with a name for the method called Amphiplex, the amphibian amplexus method. So inducing amplexus and spawning and what have you. So the Amphiplex method. In our original paper, already 11 years ago, we were able to do this in 4 different frog species. Here's the very first tadpole, leopard frog tadpole that metamorphosed following induced spawning with this method. The Pac-Man frogs are shown here, so Tophris ornata and Crenwelli. Massive declines in the pet trade, illegal and legal. So we were able to spawn these animals and study the tadpoles and learned that these tadpoles actually sing. Here's a fossorial species from Argentina, Odontophrys americanus. We took advantage of them emerging and were able to induce spawning. The method is adaptable to large-scale production, and here's a study done in these large tanks outside in the spring breeding season. You collect the animals, you put them together, and controls. This is 16 pairs, 16 females with 18 males, and none of them will spawn in captivity when you inject them with saline. If you inject them with the Amphiplex mixture, which is GnRH and the dopamine antagonist, you get 89% of them to lay eggs. Here's just another version of it with a more difficult chemical to work with, domperidone. It's not water soluble, and it doesn't give you any advantage. So stick with the easy method. Get 16 out of 18 to spawn. We estimated it to almost 3,000 eggs per egg mass, which means 2 people, myself and my wife Leslie, on a Sunday afternoon, injected them all, and about 3 days later, we produced 82,000 eggs. Highly synchronized and quite easy to do. So this is the way to go for large-scale production. So the Vancouver Aquarium saw these results, and they were trying to breed the frog that they have over there, which is a northern leopard frog, but it's virtually disappeared from British Columbia and Alberta, not doing too well in Washington State either. So how can we use this method to induce spawning in their captive animals and try to reintroduce? And this is going on, and it seems to be working well. They fly the tadpoles into the interior of British Columbia, away from the invasive bullfrog, and they're starting to have a reestablishment of the colonies. So now what's important is sometimes you need to encourage the animal to have the full LH surge. So there's a very important concept called GnRH priming. It's very much like priming your pump. You get the water up, but you don't let the water release until when you want it. So priming is really important in many, many animals, and we've used this in a lot of different species. And I'll show you some experimental data in a minute. So the idea is to get the pituitary sensitive to the Amphiplex mixture or to another hormone. So you give a low dose of GnRH, which just gives a little bit of increase and gets the pituitary ready to release a lot more LH. So a day or two before, you inject a low dose that won't induce spawning. The next day, you give the full dose, which will give you the full LH surge, and you get eggs and sperm release a few days later. So we use this for out-of-season breeding or for difficult animals. And here's some data. This is published in our second paper, which is also open access online. This is the one to follow. Very simplified procedure described here. So here's the normal mixture out of season. So we captured animals under the ice, brought them into the lab, kept them in the cold and in the dark, warmed them up, and inject them. And we get about 40 or so, 3 out of 7 pairs, released egg masses with a low fertilization rate. Do the same thing with another group with the priming, and you get the same as we got in season. 89%, so 11 out of 12, and a very high fertilization. So out-of-season protocol can work very well with several species. Now, what about other animals? So this is a really great story, the giant salamanders. The North American species is the smallest, Cryptobranchus eleganensis. Maybe a big one is around 5 kilos, a big male. But Andrea japonicus and dividianus, and new species that seem to be discovered, or subspecies, they can be enormous. This animal can get up to 50 kilos. So we started 10 years ago a collaboration with Dale McGinnity at the Nashville Zoo. They had a lot of interesting data on oocyte development in captivity, and here you have a beautiful ultrasound image. The advantage of a big amphibian, of course, is that you can see oocyte development in a living animal. You can also see the testicular structures and the epididymis full of sperm. You can measure it, and you can actually know when the animal is going to release their sperm or their eggs by ultrasound. So this was very interesting, and Dale had no luck getting sperm or eggs out, so he called me, and I went down, and we induced egg release and sperm release in this animal for in vitro fertilization. So here you have a sperm collection. They just squeeze it out, fills up this jar here. Here's Robert Brown, who's very much into amphibian sperm, looking at the sperm here, and here's a photo of the Hellbender sperm. So, put sperm and egg together, what do you get? Well, let's pull the eggs out first. So they've ovulated, and you just pull the string of pigmentless eggs out. So the animal's ovulating as you're doing this. And then you put them in little baking dishes with a particular composition of salts that should encourage in vitro fertilization. And this has been very difficult. We don't have great success, but we have success. And the very first animal that resulted from this was 1 year old in 2013. So you can do the calculation. But here this animal is still alive today. The very first Hellbender produced by in vitro fertilization in 2012. So, 9 years ago. So in 2013, this is the 1 year old, I went down for the birthday party. It was a great success. Here's some pictures from 2013. These are beautiful eggs, pigmentless. You can see them developing here from in vitro fertilization. So the first short description of some of the methods has just been accepted, and Dale's article will be published in Reproduction, Fertility, and Development shortly. Okay, so let's look at a couple of nice photos and other species, not an extensive list, but a couple of other species that have been induced by this method. In the Puerto Rican crested toads, in Nashville Zoo and Fort Worth Zoo, a single injection, both males and females, induces amplexus. They lay eggs, the tab holes develop, and you can reintroduce them. So in the example of the Nashville Zoo, McGinnity and his team injected the animals, they grabbed the eggs, and they flew them to Puerto Rico, put them in these ponds in the middle of the forest, let them metamorphose, and they walk out. So Will they survive and increase the population is the next big question. So you can induce spawning then can be used to collect gametes, so sperm for cryopreservation or for in vitro fertilization, or you can get the developing eggs and fly them to the site or drive them to the site. So it's a management tool. The timing is very important. Now, more close to home is the Boreal chorus frog. So this animal, a big one, is about 1 gram or 1.5 grams. It's called Rainet Faugrillon in French because it sings like a little cricket. In the province of Quebec, these only occupy about 10% of their original territory, so the peripheral northern population is starting to show some signs of stress. So we're working with the Environment Canada people and the Ministry of Flora and Fauna of the province of Quebec for captive breeding at the Biodome in Montreal for reintroduction in a site near Montreal. It's going extremely well, and hopefully I can report on some results at another time. Here's just a couple of nice photos. Here's an animal that's not endangered or not particularly hard to breed, Ranella fernandesi, a buffonid from Argentina, but you can induce them to spawn, so you get the eggs when you want them. This one, the Montevideo tree frog, Ipsiboas pulchellus. It's a really important, very abundant animal, important in the food chain. They have some particular behaviors that are interesting to study, and we've been able to induce spawning with them. This is work with Carolina Salgado Costa and Guillermo Natali. And we have had some success and some failures as well. So there's something we have to learn about these animals and the timing of their spawning events. So these methods can be used to as well. Here's the Panamanian golden frog. Many institutes are trying to breed Alitopus species, and here's Zeteki. This is Gina de la Tonga's work with Pierre Comazzoli at the Smithsonian Institute. This article was published a few years back, and it's looking at sperm release because what Gina and Pierre are trying to do is develop optimized methods for collection of sperm for cryopreservation and biobanking of this critically endangered animal. So here is sperm concentration and time post-injection with various doses of GnRH and HCG here. So these are different doses of HCG, GnRH and HCG. And here's a combination, the highest dose of GnRH plus the dopamine antagonist. So this is the Amphiplex method here. I think you can agree that you get more sperm, at least 2 or 3 times more, with a single injection of a mixture of GnRH and dopamine antagonists compared to the GnRH alone, and it's much better than HCG. So there's a lot of interesting things you can do by manipulating the stimulatory and the inhibitory systems controlling LH release and then controlling gonadal function, obviously. So the involvement of dopamine is looking important, lots to do. There's some good evidence. I would call it preliminary in some ways, and much is missing. When to inject, what's the mechanism? So this is one of the major issues to address in future research. Dopamine may not be important in all species. That's what we found in fish, but it's certainly important in a lot of species. Which GnRH, which dose, when to inject, it depends on the purpose of the experiment. So lots of work to do with the various forms of GnRH. I think this would be a very fruitful PhD project. And then, of course, are those tadpoles healthy? Till now, we have good evidence that they are. We've reintroduced the Boreal chorus frog into one site in 2016, and they're still singing. So these are healthy animals. But there's lots to do to understand how many survive and do you reestablish the population. Now, one thing is for sure is that amphibian endocrinology lags mammalian and fish endocrinology by 40 or more years. We are really, really way behind in terms of our research. There's a lack of basic knowledge on many of the systems. So in the last 2 slides, I'm going to show you some complex figures just to illustrate what we do know and where we may go in the future together in collaborative research. So here's kind of what we know. Some of it's speculative. It will be published this year in a book chapter and a book all about amphibian reproductive technologies that Amy Silla and Andy Kuba and others have put together. Harold Heatwall as well. So here we have what we know about neuroendocrine control of amphibian breeding. This is the hypothalamus in the brain, and factors communicate with the pituitary gland, and the pituitary gland then releases LH and FSH to act on the ovaries. The pituitary also releases prolactin from lactotrophs, and the involvement in reproduction needs a lot of exploration. I think there's a fruitful angle here as well. So what are the factors in the brain that can provoke LH and FSH release to give us adequate courtship and spawning, egg release, and sperm release? These are the big questions. So there's multiple forms of GnRH that have yet to be explored, and there's other factors, other neuropeptides that may be involved as well, and I just illustrate that here with the question mark. Now, one thing I haven't talked about at all today is the role of stress hormones. The amphibian adrenal gland is called the entorhinal. Corticosterone is released and causes courtship behavior in newts. It inhibits courtship and spawning behavior in newts. So you need adequate stress hormones to be normal, adapt, and metabolize, but too much can regulate and inhibit your spawning. So this is one of the big problems in captive breeding, figuring out how to maintain corticosteroid levels to the right level and not produce too much that would inhibit reproduction. So lots of things to go for in the future. Now, one thing I discovered recently is that there's so much to be understood in the ovary and the testes of amphibians. Now, this is in the chapter that is in press as well, and many of you will know that surrounding the oocyte are thecal and follicular cells, and this is the main site of steroidogenesis in the classic mammalian model. Well, we have forgotten very important data that the oocyte itself is a source of steroids for ovarian and oocyte development. Super important. So you need healthy oocytes inside healthy ovaries, basically. So if I just take a moment to illustrate this. So the type of cell that really produces the key precursors to estrogens, testosterone in particular, is the thecal cell. So luteinizing hormone acts on the thecal cell to produce testosterone. It's exported. The follicular cell that neighbors it is going to take up the testosterone. It can also synthesize testosterone to some degree. And then testosterone gets converted to estradiol, or to something called DHT, dihydrotestosterone, which is the androgen in amphibians. It's not shown here. FSH encourages this. So proper estrogen production, whether estrone or estradiol, is really important in both males and females, but in particular females, because estrogens act on the liver to produce the egg yolk proteins phylogenin, which are released into the blood and captured by the growing oocyte, making sure that they mature and that they're healthy, and that the larvae will have adequate yolk for early development. Now, the oocyte itself can take up steroids, and it can release them, and by diffusion, go back into the follicular cell and act as precursors for more steroids. So this is something that's totally open in terms of amphibian reproductive technologies, how we can manipulate this to get better eggs that develop better. I would like to thank you there. Again, to emphasize, this is just touching the surface. And if you have any questions, feel free to contact me. If you find any of this interesting and you want to explore collaborations, please email me. If you want help in any way, please do not help to contact me, and myself and my graduate students would be happy to assist in any way. Enjoy the various workshops, and I'd like to thank the organizers for putting this on. This is an extremely important event, and I thank you all for your attention and for putting this together.